Hello. In this video I will talk about an interesting biochemical test which is starch hydrolysis test. Before going to the main content of this test, let me introduce with this test. Amylase is an exoenzyme, and what is an exoenzyme? The enzyme which acts outside the cell. So produced inside the cell but acts outside the cell. So amylase is an exoenzyme which hydrolyzes starch, and starch again is a polysaccharide. So amylase breaks starch into maltose, which is a disaccharide, and some monosaccharides such as glucose. So these disaccharides and monosaccharides, once it is formed outside the cell, enters into the cytoplasm of the bacterial cell through the semi-permeable membrane, and thereby it is used by the endoenzymes. Starch is a complex carbohydrate composed of two constituent amylase, a straight chain polymer of 200 to 300 glucose units, and amylopectin, which is a large branched polymer with phosphate group. So amylase production is known in some bacteria, but it is well known in fungi. And amylase commercially produced from various aspergilli. They are used in the initial steps in several food fermentation processes to convert starch to fermentable sugars, and also used to pre-digest the food for young children. It is used to clarify fruit juices in the manufacture of corn and chocolate syrups. The ability to degrade starch is used as a criteria for the determination of amylase production by a microbe. So as I have mentioned, amylase cleaves starch into maltose and some monosaccharides such as glucose. Maltose is a disaccharide. So after these sugar moieties enter into the cytoplasm of the bacterial cell and are used by the bacterial endoenzymes. So amylase is produced by some bacteria, but it is obvious in fungi. So this test will determine the ability of the organism to produce amylase, which will ultimately break starch or cleave starch or hide starch. So as it is clearly visible from this picture, the ability to hydrolyze starch is used as a criterion for the determination of amylase production by a microbe. It is tested by performing the starch test using iodine solution as an indicator. So we will use iodine solution with the medium as an indicator. Starch in the presence of this iodine produces a dark blue coloration, and after the bacteria or the microbe is inoculated, if it is positive test, a yellow zone around the colony in a blue medium indicates amylytic activity. Now with the requirements, we need slant cultures, starch agar medium, grams iodine solution, sterile petri plates, dropper, inoculating loop, and Bunsen burner. Procedure: prepare starch agar medium, cool it at 45 degrees centigrade, pour into the sterile petri plates for solidification, label each with the mark of the isolate to be inoculated, a single streak inoculation of each isolate into the center to be done, incubate for 48 hours at 25 degrees centigrade in an inverted position. After incubation, flood the surface of the plates with iodine solution using a dropper for 30 seconds. Pour off the excess iodine solution. No need to remember this procedure. You will follow this procedure while doing practical.